Hi, our topic is the molecular mechanisms of antibiotics, more specifically penicillin. My name is Prissy.
My name is Lillian.
And my name is Judy. So, over 1 million people per year suffer from tuberculosis, pneumonia, and strep throat, all of which can be treated by antibiotics. So, antibiotics target bacteria instead of viruses because antibiotics have methods that target specific bacterial mechanisms and components like the peptidoglycan cell wall, something that viruses do not have. So, antibiotics are needed because the threat of highly resistant pathogenic bacteria is increasing. Antibiotics inhibit the growth and development of bacteria through different mechanisms while preserving human cells. By understanding the chemical structure and molecular function of specific antibiotics in the penicillin group, we can further antibiotic research.
We will go through the 5 antibiotics mechanisms of action. The first one is inhibiting an enzyme. These antibiotics inhibit enzymes involved in bacterial cell wall synthesis, protein synthesis, or nucleic acid synthesis. They do this by changing the shape of the enzyme, which inhibits their ability to construct the cell wall and causes the cell wall to break. The enzyme targets include transpeptidases, topoisomerases, RNA polymerase, and peptidyltransferases. An example of an antibiotic that does this is actinonin. The second mechanism of action is interfering with cell membrane permeability. One group of antibiotics, polymyxins, disrupts cell membrane integrity of gram-negative bacteria by binding to membrane phospholipids. 2 polymyxins include polymyxin B and polymyxin A. The second group, ionophores, are active against gram-positive bacteria, which lack outer cell membranes, and 2 examples of ionophores are monensin and valinomycin. The next mechanism of action is interfering with cell wall synthesis. These antibiotics weaken the peptidoglycan scaffold within the bacterial wall, which does not affect cells of a mammalian host, because mammalian host cells do not contain a peptidoglycan cell wall structure. An example of this antibiotic is vancomycin. The fourth mechanism of action is interfering with protein synthesis. One class of antibiotics, aminoglycosides, block bacterial protein synthesis by interfering with the processes at the 30S or 40S subunit of the 80S bacterial ribosome. An example of aminoglycosides is hygromycin B, which binds to a single site within the 30S subunit that distorts the ribosomal A site and induces a misreading of aminoacyl tRNAs, as well as preventing the translocation of peptide elongation. And also, tetracyclines prevent binding of aminoacyl tRNA. The final mechanism of action is interfering with DNA synthesis. Quinolines, for example, interfere with bacterial topoisomerase II and not mammalian topoisomerase. And some antibiotics interfere with RNA synthesis by inhibiting RNA polymerases.
So, the history of penicillin starts with Alexander Fleming. Penicillin drugs are all in the same family that come from a fungus called Penicillium. So Alexander Fleming was a bacteriologist at St. Mary's Hospital. He had a lab in London and returned from vacation to find that the staph bacteria had been contaminated with Penicillium notatum. Other scientists already knew of that finding, but they did not realize its significance. So Fleming noticed that the bacteria close to the mold had lysed, while the bacteria further away from the mold was fine. He then grew fungus and treated bacteria similar to the staph bacteria in order to replicate its effects. In March of 1929, Fleming concluded that fungus produced something that killed the bacteria and named it penicillin. In early 1941, Albert Alexander was the first person to be treated by penicillin after suffering from a severe face infection that caused the sepsis. The penicillin was effective, but he ultimately died from a limited supply of the drug. The widespread use of penicillin starts from an Anglo-American penicillin project, which was started after penicillin was proven to be effective and powerful. The project aimed to provide antibiotics for the Allies in World War II. In 1945, the structure of penicillin was explained through X-ray crystallography by Dorothy Crowfoot Hodgkin. In that same year, penicillin was made available for the public, and not just for military use. In 1957, the first total synthesis of a member of the penicillin family started with the penicillin V group. Today, the penicillin V group is used to treat many bacterial infections such as pneumonia, scarlet fever, and throat infections. Today, there has become a widespread resistance against penicillin. Easy access to antibiotics is likely to have promoted overuse of these drugs. The diagram shows how antibiotics kill many of the bacteria but not all of them. These resistant bacteria then go on to reproduce, producing more resistant bacteria. The resistant bacteria soon become the majority of the bacteria, and the bacteria can then transfer their resistance to other bacteria, which strengthens resistance against penicillin as a whole. The use of antibiotics for viral infections also increases resistance, as it should only be used for bacterial infections.
For the core structure of penicillin, it has the molecular formula of RC9H11N2 or 4S, where R is the variable side chain. So if you change the R group, you change the function of the penicillin. All penicillins have intermolecular forces of London dispersion forces, dipole-dipole forces, and hydrogen bonding. So all penicillins are beta-lactam antibiotics, which means that they contain a beta-lactam ring, which is highlighted in red in the two diagrams over here. So what a beta-lactam ring is a four-membered lactam. So the nitrogen atom is attached to the beta carbon atom relative to the carbonyl group, which is the C double bonded O. Most beta-lactam antibiotics work by inhibiting cell wall biosynthesis in the bacterial organism, which is one of the mechanisms of actions of antibiotics, as mentioned before. Beta-lactam antibiotics are also the most widely used group of antibiotics. So penicillin targets specifically gram-positive bacteria. This is because gram-negative bacteria have lipopolysaccharide and protein layers that surround the peptoglycan layer of the cell wall, which prevents the penicillin from attacking. Further research has allowed some penicillin to attack some gram-negative bacteria, but that research is still in development. So for the molecular mechanism of penicillin, as we said before, gram-positive bacteria possess a thick cell wall composed of cellulose-like structure, sugar polymer covalently bound to short peptide units in layers, which we said was the peptoglycan layer. So what peptoglycans do is that they create a mesh-like structure around the plasma membrane of bacterial cells, which increases the strength of the cell walls. When bacteria multiply, small holes open up in its cell wall as the cell divides, with newly produced peptoglycans filling up the holes to reconstruct the walls. So penicillin then binds at the active site of the transpeptidase enzyme that cross-links the peptoglycan strands. So D-alanine is the backbone of the peptide chain of the cell wall. Penicillin mimics the D-alanine. As you can see in the diagram, the structures are very similar, which allows penicillin to inhibit the target protein. This eventually leads to cell lysis. Cell lysis is the breaking down of a cell. Penicillin prevents bacteria from closing holes in its cell wall, which prevents the bacteria from multiplying. Bacterium is then so filled with solutes compared to the surrounding solution that the water rushes in, and without a full cell wall to support the bacterium, it pops from the pressure.
So there are many examples of penicillin, one of which is amoxicillin. It is used to treat bacterial infections such as skin infections, strep throat, and pneumonia. Its chemical structure consists of a beta-lactam ring and includes an amino group and a carboxyl group. It has a molar mass of 365.404 grams per mole. It can donate 4 hydrogen bonds and accept 7 hydrogen bonds, and it has a formal charge of 0. Its molecular function is to bind to penicillin binding proteins, or PBPs, on the inner membrane of bacterial cell walls, which weakens the cell wall and eventually leads to cell lysis.
The next example of penicillin is oxacillin. It is used to treat infections caused by penicillinase-producing staph bacteria. Its chemical structure, like all penicillins, contains a beta-lactam ring. Its molecular mass is 1.437 grams per mole. It can donate 2 hydrogen bonds and accept 7, and its formal charge is 0. Its molecular function is to bind to penicillin-binding proteins in the bacterial cell wall, blocking the synthesis of peptidoglycan and causing cell lysis.
Another example of penicillin is piperacillin. So it is used to treat pneumonia and skin, gynecological, and abdominal infections caused by bacteria. Its chemical structure is also a beta-lactam ring, and its chemical and physical properties are that it has a molecular mass of 517.557 grams per mole. It can donate 3 hydrogens and accept 6, and it has a formal charge of 0. As stated before, the molecular function of piperacillin, as all penicillins, is that it binds to penicillin proteins, penicillin binding proteins on the inner membrane of bacterial cell walls, which weaken the strength and rigidity of the cell wall, leading to cell lysis.
So the last example of a penicillin that we have is ampicillin. It is used to treat bacterial infections such as ear infections, bladder infections, gonorrhea, and E. coli infections. Its chemical structure also contains a beta-lactam ring. It has a molar mass of 349.405 grams per mole. It can donate 3 hydrogen bonds and accept 6 hydrogen bonds. It has a formal charge of 0. Its molecular function is also to bind to penicillin binding proteins on the inner membrane of bacterial cell walls, which weakens the cell wall and eventually leads to cell lysis. However, it is stable against hydrolysis by beta-lactamases, making it effective against gram-positive and gram-negative infections. In conclusion, antibiotic development is important because of the rising issue of antibiotic-resistant bacteria. Antibiotics are effective because they target significant structures in bacteria and inhibit the bacteria's growth and development. By understanding the molecular mechanisms of antibiotics, we can further our understanding, which can ultimately lead to revolutionary drug development that attempt to target the detrimental nature of bacteria. Thank you.